antibiotic apocalypse happens when bacteria become resistant to antibiotics, which previously treated the infections caused by these bacteria. Antibiotic resistance has become one of the biggest threats to global health, as current treatments will no longer work. Resistance has now been clearly linked to antibiotic overuse. Antibiotics kill susceptible bacteria in animals while allowing resistant strains to thrive. But how exactly do these bacteria become resistant?
Bacteria are prokaryotic organisms that can be transmitted via person-to-person contact, food, contaminated water, insects, and fomites. Additionally, bacteria can spread through coughing, sneezing, and exposure to bodily fluids. These are dormant structures which are extremely resistant to hostile physical and chemical conditions such as heat, UV radiation, and disinfectants. Bacteria have been dated to be on the Earth as far as 4 billion years ago, when oxygen appeared in the atmosphere. During those periods, oxygen levels were scarce, and the first type of bacteria found in fossils have been dubbed cyanobacteria. Bacteria are classified into 5 major groups: spherical, rod, spiral, comma, and corkscrew. Bacteria can exist as single cells, in pairs, chains, or clusters. Bacteria are classified as prokaryotes. This means that they have unique characteristics, such as their unicellularity and the lack of membrane-bound organelles.
The use of microorganisms to treat microbial infections in ancient China, Egypt, and Greece has been well documented. However, the modern era of antibiotics began with the discovery of penicillin by Sir Alexander Fleming. After an uncovered petri dish near a window was contaminated with mold spores, Fleming noted that the bacteria surrounding these mold colonies were dying. This mold was identified as a member of the Penicillium genus. Penicillin was largely effective towards treating infections during World War II until the emergence of resistant strains in the 1950s. This signaled the start of an arms race, and bacterial resistance has been encountered in nearly all antibiotics developed since. The antibiotic pipeline is now drying up, leading us to believe that we may never win this war against microbes.
How do bacteria reproduce? And what exactly is their method of resisting drugs? Bacteria reproduce via binary fission, a process by which a single cell divides into 2 identical daughter cells. Nearly 80% of all antibiotics sold in the US are used in animals to promote growth and to prevent infection. The production and sale of antibiotics is not regulated in all countries, which provides cheap public access to these agents in large quantities. Sir Alexander Fleming warned the public regarding antibiotic overuse over half a century ago. In addition, inappropriate prescribing of antibiotics further contributes to the overuse of these medicinal agents and development of resistance in bacteria. Bacteria have genetic plasticity that allows them to respond to an array of environmental threats, including antibiotics. Antibiotic resistance arises from 2 major mechanisms. First, the acquisition of foreign genetic material that codes for resistance. This typically occurs through horizontal gene transfer, which is a predominant driver of bacterial evolution, as well as acquired antibiotic resistance. Second mechanism is mutational resistance. Resistance through mutation is random. A subset of the bacterial population randomly develops mutations in genes that affect the activity of the drug. This results in bacteria cell survival in the presence of the antibiotic. Once a resistant mutant emerges, the antibiotic eliminates the susceptible population and the resistant bacteria predominate. The surviving bacteria subsequently proliferate to produce further bacteria who are also resistant to the effects of the antibiotic. Mutations resulting in resistance alter antibiotic action by the following mechanisms: modification to prevent target antibiotics, activating efflux, changing metabolic pathways, and decreasing uptake by changing cell permeability. Bacteria may acquire external genetic material through 3 mechanisms. First, transformation, or the incorporation of naked DNA into their genome. Second, transduction, a process where foreign DNA material is incorporated into a bacteria through a vector. Third, conjugation, a process by which there is a transfer of genetic material from one bacteria to another via sex pili. Superbugs are still a threat to us as humans, but for the bacteria, they are truly a benchmark of modern evolution.
Antibiotic resistance can be minimized by preventing overuse of these agents by only taking antibiotics when prescribed by a healthcare practitioner, always completing the course of the prescription, and never sharing or using leftover antibiotics. The agriculture sector can minimize the use of antibiotics by vaccinating animals and adopting sustainable methods such as good hygiene as well as stress-free handling of animals. Additionally, healthcare workers must promote the appropriate use of antibiotics by dispensing the correct antibiotic for an appropriate duration and only prescribing antibiotics when truly necessary. The antibiotic apocalypse may still be underway, but as we have seen, there is still hope if we follow the necessary precautions.